for coming. So yeah, thanks Sam for the invitation, and I'm going to talk, as you were saying, mostly about transcranial alternating current stimulation. So I don't know if you're familiar with different forms of non-invasive brain stimulation. So people have been using TMS, transcranial magnetic stimulation, for quite a while now, and there has been sort of growing interest in using current forms of non-invasive brain stimulation. So stuff like tACS, so transcranial direct current stimulation, alternating current, random noise. There's many different modalities, and I'm going to talk about how we've been using this to understand cognitive function, and how we've been putting this together with Bayesian optimization to kind of like tackle some of the issues associated with using brain stimulation to modulate cognitive functions. Okay, so the talk is essentially divided in two parts, and I'll spend a lot of time here. So the first piece is on challenges and new strategies to improve reproducibility in brain stimulation experiments. The second one is how you can use this approach, so neuroadaptive Bayesian optimization, because you guys RIOT Club, so to broaden even more open research practices and improving reproducibility. And this will be quite short, but I'll give you some nice references where you can check more information. Okay, so going back to transcranial alternating current stimulation, tACS. Why am I interested and others in use this to modulate cognitive function? So the reason for this is that we have this sort of idea that you have a network of regions that is necessary to support cognitive function. You have different brain states. Brain oscillations seem to not be just an epiphenomenon, so they seem to be associated with cognitive functions, and people have associated different frequency bands with particular cognitive functions. Like one example is theta waves between something 4 and 7 to 8 hertz that people have associated with working memory again and again, and this has been shown in animal models up to humans now with invasive recordings. If you guys have questions at any time, just stop me, okay? I don't know exactly where you all are, so just ask away. So the idea here was really, okay, if these oscillations are important and the distribution of these oscillations is somehow important, okay, where they occur, if they are across frontal and parietal cortexes, for example, if this is important, and if is this indeed associated with behavior, then it should be possible to use an external oscillator to modulate your internal oscillators. So this is like the basic idea for why we want to use alternating current stimulation instead of just a direct current in the brain. Okay? So the appeal here is really that you can tune your alternating, your external modulator to a particular frequency that you are interested in. And hopefully by doing this, we can go a little bit, can go beyond correlation, and we can causally investigate this brain behavior relationship. So if we can shift this brain activity in particular frequencies, and we shift which show differences in behavior, then we are showing that actually these oscillations are important, the regions where they occur is important, and we're able to tweak that. And this is attractive because alterations in brain activity are seen, alterations in brain networks are seen across all sorts of neurologic, psychiatric disorders that you can think of. And there's not many available treatments for cognitive function. So if you can use something like brain stimulation, which seems less invasive than other approaches, then you potentially combine with other strategies, you can have something else to help patients suffering from cognitive disorders. However, as Sam and I were talking in the beginning, there's a lot of controversy around the replicability of this. So there's been quite a few papers that are quite polemic in the field, saying, well, there's actually no cognitive, actually brain simulation in this form, tACS, doesn't show any cognitive modulation. And people were forced to sort of stop and think a little bit better about the way they're conducting their research. And the problem here is that this is sort of the traditional approach for when you want to do a brain stimulation study, particularly if it's a cognitive one. So you have some a priori idea about what regions you want to modulate in the brain. And this can be from your own research, so you collected this data, you have some sort of idea what you want to do, can be from a meta-analysis. You might have an idea of what frequency, here talking about alternating currents or tACS, you would want to modulate. This is not always obvious. You can think about, okay, what I want to do is actually, I want to modulate connectivity between different regions of the brain. So I know I don't want to modulate just this region, because when I look at the pattern of activity for a particular task, or I know that, well, I want to modulate in this case sort of more parietal and frontal areas. So you can think about putting different electrodes in different positions and modulate the phase between them, okay? The intensity between them. So a lot of things that you can think about that you can modulate. And the intensity, as I was saying. So typically you would do this sort of at a group level, okay? And then you get your participants coming into the lab, you have them doing a task, you apply brain simulation at the same time, and hopefully you have one more control that's not just sham. So sham is applying brain simulation for a short period of time. So people typically do is that they have an intervention for like 10 minutes, 20 minutes, where people receive brain simulation continuously for that amount of time, and then your control is 30 seconds of stimulation in a different day. That's the most typical sort of approach. And this has been shown again more and more that carries many problems. So hopefully you have an additional control, like an active control. It can be a different frequency, it can be a different montage, so you can think about what would be the best control for your experiment. And I would say that this, with something like this, you face two main challenges. One is variability. So it's really hard to know what the effects of simulation are in brain function if you don't have, if the only thing that you're recording is behavior, if you don't have any other modality that will tell you whether, well, my a priori assumption was that I was modulating frontal activity, did I do that or not? And the other big challenge is inter-individual variability. So here, and this is particularly very, very true for brain stimulation. This has been shown in TMS studies, so it's a known fact that the stimulation parameters may vary a lot across individuals. So this can be the intensity. If you think about frequencies and you've recorded EEG, we know that people have, they vary their resonance frequency for a particular task. And this becomes even worse if you want to apply it to patients, where the individual variability might be even bigger. Okay, so how have we been trying to tackle these two challenges? That's what I want to talk about. So to tackle the first challenge, the way we've been doing this is to combine brain simulation with a simultaneous recording technique. So let's see what actually the simulation is doing to the brain. And I'm going to talk about MRI, how I've been using magnetic resonance imaging to do this, and why. So the first, I think, main attractive is that you can record brain activity, brain function, in the absence of artifacts. So in the beginning I was telling you about frequencies, right, and how we can record those with EEG. The problem is that your EEG is acquiring is recording brain activity that has in the microvolt range. Okay, that's millivolt to microvolt range is what you can get. And then you're imposing at least 1 millivolt on top of that. And people have tried to recover the signal, and this has been shown to be very, very difficult because you have complex interactions between physiological signals like heart rate, respiration, and the imposing current that you're putting on top of it. So currently, I would say there's a very good solution to do this. There's some strategies that you can use, but I would say it's really hard to be sure about whether what you recover, particularly in the frequency that you stimulate, is a true endogenous signal or is some residual of what you imposed. So other advantages with MRI is that you have this high spatial resolution, so many times you interest not only cortical regions but also subcortical regions. We know that brain stimulation, when we apply in this non-invasive sort of way, the current will flow wherever it can. So sources like CSF, so the current will go to the path of less resistivity. So don't mistake yourself, don't fool yourself. This is not a sort of like very constrained application of a current in a particular area. So if you want to do this, you need to use other technologies. So the current will flow to the path of less resistivity, and you will see activity in subcortical areas as well. And the advantage is that once you have someone in the scanner, you can acquire a bunch of modalities that might allow you to investigate the same problem through different ways. So you can do spectroscopy, you can do DTI, you can look at anatomy, so volumes of grey matter, white matter, they should have an influence on how the current spreads in one's brain. And quite interesting, people have developed current density images which allow you to map how the current flows in the brain. So the current that you're posing, you can get a sort of map of that per individual. And of course, yeah, there's disadvantage, right? So false a surrogate signal of neural activity, and you can't get all the frequencies that you would get with EEG. So I was talking about teeth oscillations. I can't get this directly from MRI because of the sampling rate that I have. However, people have shown there's some relationship between at least the envelope of EEG rhythms and bold oscillations that we can recover at the frequencies that we can't. Okay, so how does one do this? It's actually very simple, and you don't need a lot more. So there's a number of companies that sell MR-compatible stimulators. They're not difficult to do yourself. Actually, this is a 8-volt battery sort of thing that you're using. You're not going to put in people currents that can harm them. So there's actually, like, not to do this in the scanner, but quite a big community of people doing this themselves and building stimulators, and this is a reality that, well, another conversation. But you have your stimulators, and when you want to put them in the scanner, what you need to think is how you route the cables through the scanner. So if you've done anything where you put something inside the MRI, you will know that you need to be careful the way that you route the cables. You need to be careful about the length of the cables so they can work as an antenna, and they can generate currents on top of the current, in this case that you're putting. So different companies have come up with different solutions for this. This is an example of what Neurocon uses. So there's 2 filter boxes, and I had to slightly modify this so I wouldn't have noise in the scanner. And if this is something you want to do, you're very interested, I can explain how it changed and included another RF filter. But yeah, you don't need much more. Of course, people charge you much more because anything that goes into MRI, much more expensive. And the other thing is, in the scanner, you might not want to just stimulate throughout whatever 10 minutes. You might want to be a bit smarter in the way that you design your combined stimulation and MRI study. So you might want to do like a block design, for example. where you'll get a better signal to noise ratio in terms of the physiology, and you can have more conditions, and you can compare across conditions. So you can do the thing that I was telling you. Well, in the same run, you can have controls. You can have your active condition, your control, your sham control. You can play with the duration, the tasks they're using. And if you want to do that, it's again simple. You just need to get your simulators being controlled by a digital to analog converter. So they'll take voltage in and they'll output voltage out. So you can easily code something like this in MATLAB and define, in this case, any sort of like pattern that you want to stimulate. And this is sort of what we did. And this is the first study that we did in the scanner. And I told you about theta. Theta is involved in working memory, so it's this frequency that people have associated again and again with working memory. People have also shown that working memory, you have this sort of like, when you look at working memory, patterns of activity, you have this really nice frontal parietal network being involved in working memory tasks. So we were interested to see whether if you put electrodes on your frontal parietal cortex, whether we could modulate people's behavior in a working memory task. So whether we can make them either better or worse in a task, and we did this by varying the stimulation parameters that we would apply in frontal parietal electrodes. And this was really nice. So we saw that depending on the way that we apply stimulation, so what we did here is that we had this, so this is EG coordinates, if you're familiar with this. So like F4 will be in the frontal right side, P4 parietal. We had a common relative here in T8, and what we did was we varied if the current that we're injecting here was in phase or out of phase. And what we see is that when we injected in phase, we saw an increase in activity in this frontal parietal network that is involved in working memory. We see that this was related with people being faster in the task. And the larger the increase in bold that we put here, the better they were in this, the faster they were in this working memory task, And we saw that if we inject current in a different way, we had this sort of like default mode DMN sort of pattern. So really striking changes in physiological response depending on the current that you're injecting. And we show that you actually can modulate as well brain connectivity. So we could increase the connectivity between parietal and frontal regions using one particular setting, and increase between parietal and occipital regions using a different setting. And this is really nice. So we didn't know why, but last week just something came out on Nature Communications. So Alex Alpitz did a study in monkeys using this configuration, and they showed that with using traveling waves, they explained exactly this pattern. So it was really nice to see that, well, someone else got the same, and they can really see the activity invasively, and they can explain these sorts of results. Cool. So this is a bit of what we did. The study had more things, and I'm happy to discuss. So we had like another control with a task control, and this was important to show that, well, this doesn't happen if the areas of the brain that you want to modulate are not engaged in the task. So if you put the electrodes on the frontal parietal cortex and your task doesn't engage the frontal parietal cortex, brain stimulation is not going to make the frontal parietal cortex engage. And the reason for that is that this is a neuromodulatory thing. So it's like, just think that someone is already in a swing and you're just pushing a little bit. You're not causing action potentials. So it's nice to have like a bunch of controls so we can understand better what we, the results that we might obtain with brain stimulation. And because this is interesting, we've now been applying it to traumatic brain injury. And this is actually, it was the initial idea of my fellowship, was to think about, okay, people with traumatic brain injury, they have these lesions in white matter tracts. And in order for regions of the brain to communicate with each other, to pass information, the integrity of this white matter tracks is important. So if the information cannot pass between one area and the other because there's some lesions, maybe we can control it in an external way. So we can use these external oscillators to control the internal oscillators. So this was like the purpose of the whole thing that I showed you before, but we had to show that, well, we could actually do this in healthy controls first. And we focus on frontoparietal networks, and we focus on working memory because patients with TBI have problems with working memory. And we did this N-back task that I was telling you about, so working memory tasks that people have used a lot. And what we observed is that, as expected, when we compare activity, healthy controls of patients with TBI, they have less activity in the frontoparietal network, and they also have less deactivation of the default mode network, and this has been shown again and again. And the good thing here is that when we apply stimulation, I had changed, it doesn't matter. When we apply stimulation, if we apply in one particular condition, we are able to recover, so patients no longer are different from controls. In another condition, we partially recover this, and the only difference that we still have is on the default mode network activity. And I was telling you, okay, it's good that we have the patients in the scanner, so we can collect more information and try to understand why is this the case. Patients vary a lot in a way, much more than healthy controls, in the way that they respond to stimulation, and we knew that some patients would respond and others wouldn't. So we acquired DTI, and this allows us to look at whether the integrity of the tracks was related with the effects of brain stimulation. We did spectroscopy, again, to understand whether the levels of glutamate and GABA in a particular region were associated to the way that stimulation was manipulating brain activity. And this is something ongoing, but with the DTI we have recently published, so showing that depending on the integrity of the white matter tracks, stimulation might or may not be effective. So it's trying to think about, if you want to use this as an intervention, can you stratify your patients beforehand? Can you think about who is going to benefit from an intervention? Or can you localize your electrodes in a slightly different position if the integrity of the tracks somewhere else is preserved? Okay, so now to my second challenge, which is the really difficult one. So imaging, we can figure out, lots of smart people thinking about ways of putting more stuff in the scanner, and I'm sure people will figure out ways of like cleaning the EEG signal for us to recover what's going on when we apply brain stimulation with EEG. But into individual variability, this is not easy to overcome, because this comes with so many sources. So you can think about neuroanatomy, size of your skull. If you're thinking about a patient, did he have a stroke? If you had a stroke, if you put the electrodes close to an area where you have lots of CSF, that's where your current is going to go. So thinking about all of these variables, neurochemistry, so I was just showing you, depending on the levels of glutamate, the stimulation is more or less effective. And in our case, people have shown results with GABA as well. Then you have genetics, then you have age, then you have gender. So you know where I'm going here, so it's really complicated. And my final message with this is that even if we knew all of this about the participant that you want to do an intervention, we don't know how these things interact. I have no idea. Even if we don't have a model where you can input all of this information and you can predict what the stimulation is going to do to the brain. And the reason for that is that on top of all of this, there's the dynamics of your brain. So your brain is not stationary. And I didn't show the slides in the beginning where I was telling you we had a control task when I did this with healthy controls and with a patient. And if the brain is not engaged if your brain is not engaged, if the areas that you want to stimulate are not engaged, you don't have a response from brain stimulation in particular. So, not easy to model. And then, if on top of this you think, okay, and now I have a technique like alternating current stimulation, I need to pick a frequency. And if you think about the biological range of frequency you can pick, you can think, let's say, let's just go from 1 to 100 in steps of 1. You can then think, okay, I'll have more than 2 electrodes in the brain, so I can play with the phase between them. And if you just think about like, yeah, so you go from 0 to 159, in what steps do you go? So you see how I'm going with this. So you start building a big parameter space that you have to pick in the beginning. And then what intensity? So it might be people, at least with tACS, have shown sort of like simple dose response curves where if you actually put more intensity, it doesn't mean that the brain will respond in a linear fashion. So this just becomes a sort of like combinatorial exposure. Okay, so we really need to be smart about the way that we tackle this and thinking about what other strategies are out there. So clearly, no matter how good we are, how hardworking we are, we'll never be able to map this parameter space in a traditional way. You won't have participants to do this. You might find like animal models where you can test this will take a long time. Okay, so that's where Bayesian optimization comes to play, and that's where this sort of neuroadaptive paradigms come to play. By neuroadaptive, I just mean that your paradigm, so in this case, if you can think about like the stimulation that you're inputting is changing depending on the way the brain responds to that stimulation. And people have applied this in many ways, so like open loops in the way that I can, for example, measure your brain activity, and I have, for example, a particular frequency that works for you. So if you close your eyes, your alpha frequency has a particular signature, so I can use that to stimulate. But again, your brain is changing, so none of this is stationary. And people have shown that your alpha activity also changes depending if you get tired. So you can do like some sort of closed loop. So closed loop seems better because you can adapt whatever the stimulation that you're doing depending on the way that your brain is responding. And in all the sort of like closed loop approaches, we are advocating for Bayesian optimization. This is what we are using. And the reason for this is that Bayesian optimization is particularly useful when data acquisition comes at a cost. And here, for this particular case, we have lots of costs. We have the cost of time. So if you're doing this in a scanner, how long it will take to search different space, comfort for your participant. They'll probably not be able to line the scanner for you for more than an hour and a half in a go. It'll start moving a lot, and then you can't collect more data. And also the amount of stimulation that you can give to someone. So limited the amount of stimulation that you can give, because we don't know what changes brain stimulation will cause in a sort of like long time frame. Okay, so you can think about Bayesian optimization as this closed loop approach, an iterative closed loop approach where you have 2 stages. Your first stage is the data modeling, and here what you have is a probabilistic surrogate model that is estimating your objective function. So the objective function is what you want to know. I want to know how a particular parameter of brain stimulation will impact my brain activity. Is this my behavior? Is this relationship between the two? And typically we use custom processes, and this is because they're quite flexible and they're quite versatile. And the second stage is your guided search. So the algorithm needs to propose a next step. So what is the parameter that is going to search next? What is the type of stimulation that is going to give you in the next iteration? And this is our acquisition function, so it's defined by our acquisition function, which is going to determine how exploitative or how explorative you're going to be, the big matrix that I showed you in the beginning, okay? So yeah, this closed loop approach. And now just 1D sort of example of how this works. So here in this dotted line, we have our objective function. So we have some sort of idea of how, we have some prior beliefs about how our objective function, what is its landscape, how it is. So I can have an idea about, well, I think that particular frequencies will elicit a certain behavior. I can think that particular frequencies will act in a linear way, or the intensity will be linear in terms of the brain activity, or I can think, well, actually I might have some sort of like a Gaussian distribution. Then we have our surrogate model, which is sort of like a model of a model. It's trying to model the objective function. And what the surrogate model does in the beginning of the experiment is to use all the samples that it has. And this surrogate model is constantly being updated. So as you acquire more data, the surrogate model is updated to all the points, all the data that you have until that point. And it's gaining more and more confidence, okay, about whether it explains well your objective function or not. And then you have your acquisition function. An acquisition function, what it's trying to do is to decide what is the best next point to explore in this space. So the acquisition function wants to maximize your objective function, so it's trying to think, okay, what is the most informative point that I can acquire to map this space? And these are the pink dots here. So this is a new proposed point by the acquisition function, and as you go more and more iterations, you have a better idea of you're maximizing your objective function. Yeah. So the idea is that this will be an efficient and a reliable search over an exhaustive parameter space. It will be quite efficient because we should be able to do this in a short time frame. And I want to give you an example of this applied to brain simulation. So if there are other things applied to cognitive function, and maybe Rob has already introduced you to all of that, so I'll give you an example of applying to brain simulation. So we started by doing a proof of concept study, and we started with something super basic, which was not that trivial to explain to reviewers. So we wanted to have something, we wanted to start by looking at something that we knew the ground truth. So the model doesn't know the ground truth, but we kind of have an idea what the ground truth is, and these are phosphines. So phosphines are flash-like visual percepts, so you can get those by stimulating either your occipital cortex or by stimulating your retina. So if you stimulate your retina with tiny current, you see like flashes of light. And this is known for a very long time. People have thought about approaches of using phosphines to give blind people a way of mapping the world. People thought the first time that someone saw phosphines that this could be a treatment. Yeah.
Is this the same thing as seeing stars when you get punched in the head?
You mean celebrities?
So
I mean, you probably haven't been punched, but
no,
I've
yeah.
seen stars.
And if you put your finger in your eye for a long time, you start having some. So these are properties of your retinal ganglion cells. Yes. So they are responding to
The impact.
yeah. I don't know like then all the process to the brain, but yeah, if you without the impact, if you just press your eyeballs for a bit, you start seeing those, and they have to do propensity. Yeah. So what people have seen is that particular frequencies are better of causing this flash than others. And frequencies around the 19 hertz are perfect for this. So if you take part in one of these experiments and someone shows you like 19 to 20 hertz, like a Christmas tree with lots of light. But people hadn't investigated, so this is our ground truth. We had an idea where the effects should be, but we didn't know whether phase was going to affect this. So if you had electrodes with different configurations playing with the phase. And one thing that we knew is that if we tested all the possible combinations of frequency and phases, we'll have participants in the lab forever. So our best estimate would be that in 90 minutes we could kind of like sample all the parameters once. But you normally don't want to sample this just once, so that's where we use adaptive Bayesian optimization. So the idea here is that you have a participant with the electrodes in a different configuration from here, where you ask them to do is to indicate, so they'll show, they're being shown phosphines or not, so they're shown, they just have to stare in a screen, like this could work with a white wall as well, so they just have to stare. They are shown one block with particular parameters, another block without particular parameters, and they just have to say which one they thought that the phosphines were more intense. And we're using our Bayesian optimization algorithm to then suggest a new sample and analyze this data in real time. And the reason that we ask people to compare, so say which block they thought the phosphines were more intense, is that the reason is that because people tend to be bad at doing this sort of like absolute judgments. So we're humans, we are much better at saying this better than this. Then this referred to whatever referential was stronger or less strong. So when we adapted the Bayesian optimization algorithm to be able to do this binary preferences. And this is just a snapshot of the study. So as I was telling you, participants had these two blocks where the algorithm had proposed two samples to test. People would have to rate, and we were sampling this parameter space, and we did this with a number of different voltages because we wanted to investigate a little bit more whether the configuration that we had would affect phosphines or not. But we started with this one, which is a sort of classical montage. We put one electrode here and one electrode in the occipital, and people have studied this a lot, and they show, yeah, this is how we find the 19 hertz. And we're able to do the same, so we say, okay, yeah, so we proved that our approach also works. But what happens if we start putting more complicated sort of montages where you can start having interactions between currents? So we show that, well, okay, we can still recover, so this is what you get at the end of your Bayesian optimization procedure. So each black dot is a parameter that was sampled, and in blue you have where the algorithm predicts that the probability of using a phosphine is higher, and the white dot is where is highest, so it allows us to tell us, okay, so around 19 hertz does this happen. And then in the same part, in other participants, we focus just on phase, so is there a particular effect of phase? And the reason for that is because the effects of frequency are much higher than the effects of phase. So we're able to show that not only you have an effect of frequency, but that you have an effect of phase. So if the current that you inject between two different electrodes is in phase, so 0 degree phase lag between these two oscillations, your perception sensation of phosphine is higher than is 180. And we didn't know exactly why this was the case. And computational models were really useful for this, because allow us to see that actually what we are obtaining is just an effect of simple physical interference. So when you have the two oscillations coming the same intensity, they reach the retina at a higher intensity. The retina is super sensitive to this, lots of water, so it allows this current going through. Whereas when you have 180, the current is actually cancelling out in your retinas, and that's why you have this intensity. And then, just to finish, we wanted to check whether if you use different objective functions, so if you think that your space is sort of different when you start with your assumptions on how your participant is going to perceive phosphine changes depending on the noise of your estimates. What we did was a bunch of simulations thinking about, okay, have a rate as a human that's really good at distinguishing between two blocks, have a human that's less good, and wanted to see, okay, depending on the objective function that we have, can we still recover an accurate response? And he used to say that you can play with this and trying to understand how robust your objective function is to the noise in the system. So some objective functions like the sum of squares and the common back work quite well. So this is the number of iterations that you need until you have a good idea of where you would have, in this case, a peak phosphine intensity. but others more complex might not capture that well, that response between the rate and the frequency that we're imposing. And what I'm doing now is extending this to doing to modulating cognitive function, which was the objective to start with, but it wasn't easy. So we had to figure out a way of combining this in the scanner, so doing real-time fMRI, finding a way of searching through a large parameter space in an efficient manner, so we can finally think about how we can propose a set of parameters that will work for the individual. So the idea is that you would come to the lab. We want, for example, to increase frontoparietal activity or connectivity, and we sample this large parameter space, and we'll find for you the parameters that will increase your frontoparietal activity or connectivity. And we think that this should then be a more reliable and efficient way of modulating behavior, and should be much more applicable if you have a disease. So the conclusions from this first part, and my second part is 2 minutes, I think, is that, well, to address the verifiability issue with imaging, with brain simulation, please use some imaging technique. It's not that complicated. If you just collect behavior, it's really hard to know if what you set to target is actually what you target. And there's this big issue with individual variability. This is the one that is really hard to address, and we're thinking about new ways on how Bayesian optimization can help us with this. So finally, for the RIOT Club, open science and reproducibility, and how using this sort of Bayesian optimization approaches can help even more with open and reproducible science. So the idea here is that if you have a very biased experimenter, what might happen is that you decide you experiment a space to start with, so what stimulation parameters you want to apply, and then you collect your data, you analyze it, you have your target measure, and at the end of it you have your hypothesis, and you have you write your paper. So this will be the most biased sort of way. The ideal standard now is that you have your hypothesis, you define a parameter space, you define how you're going to calculate this, what the methodology that you're going to use, you measure this, and you define the measures, you pre-register, and then you conduct your experiment, and you have your analysis. And the way that Bayesian optimization can go a step further is that you would do all of this, but you can also pre-register much more information about a large experiment parameter space, right? And you can pre-register the whole continuum between exploration and exploitation, because it's embedded on the Bayesian optimization. So before you start, you need to have some sort of idea about where is your parameter space, how is it going to be explored, and you have all this a broader range of hypotheses that you can approach, that you can test here. So you have this inherent closed loop between your experimental condition and your data analysis that you've predefined when you started. And it's locked when you're doing the experiment, so you shouldn't be able to tweak it as you go along. So yes, the idea is can combine this with pre-registration to cover this exploration phase that we also want to have as researchers, right? So we want to see what else is happening with your data, but this can help really to mitigate research bias. And there's this really nice list on everything that you can do, or how can you improve. So it can increase the specificity of the hypothesis, because you have the whole experiment space, and you can have an idea, or you can propose. So instead of just one hypothesis, you have a hypothesis about the whole parameter space, right? It includes replication by default. So normally the acquisition function that I told you about is transitioning from a place in the beginning where it's exploring different points to then when it gets more information, it's going to exploit the same points again and again. And this is out of sample prediction. You're giving a new individual sample every time. Yeah, and as I told you, expands the applicability of pre-registration, so you can pre-register this whole exploration-exploitation continuum. And I think finally increases transparency, because you implicitly are writing information about the relationship between experimental conditions into your data, and you can allow others to see this. So the maps that I was showing you, the probability maps that I was showing you where to find phosphines, those are at the group level. We can show them at the individual level, as we did in the paper. And important about the phosphines, I forgot to say, because I told in the beginning we'll need people in the lab for 90 minutes. It took us 5 minutes to find an optimum. Yes, it's efficient. But you can publish these maps, right? And what reviewers and readers can do is see exactly the trajectory that the algorithm took. And if it's all noise, it should go around and around without converging anything. And if you tweak it and if you bias, you actually can see that actually the probability of you ending up here is probably the same of you ending up here. So you can really have an idea about which participants your optimization worked for, which ones it didn't. So we had in our first experiment 2 participants for which we couldn't find an optimum because they couldn't see phosphines. So it's normally that you can't find any sort because they just couldn't see, the intensity wasn't. So you can see that, and you can see that the algorithm behaved like this in those particular cases. And of course it's finished thanking lots of people that did this. So a lot of this works from Miss Black. Romy was a PhD student with Rob Leach when we were all at Imperial, so we used to sit together and we did a lot of this work because we were all in the same space, and then lots of other people at Imperial that contribute this. I was at John Rothwell's lab when I started getting some interest in brain simulation, as Sam was saying. Ilka did the computational modeling, and yeah, so funders, so to expand this Bayesian optimization to cognitive questions. I just got an investigator from the BBSRC, so really happy to taking this through and trying to see if we can use it to improve brain simulation practices. And thank you.
We've got 5-10 minutes for questions. Does anybody have any questions?
Very interesting. My question is, I mean, with phosphines, that's very easy because you have physiological outcome here, which is very easy to determine the stimulation, whatever parameters, yeah. But with cognitive functions, it's far more complicated, right?
Absolutely.
I mean, you would have to constantly test the task, and then you have the problem of repetition effect, test, retest, no?
Yes.
How would you do that? Like if you wanted to, say, test that stimulation of this particular area improves working memory, for example.
Absolutely.
No,
So it's a great question. Yeah. So it's how do you go from your proof of concept study, and the reason we started with phosphines, we didn't know whether this is going to work or not. Mm-hmm. So we wanted to show, okay, if we know the ground truth, we are able to show something that is meaningful. How do we then get to cognitive function? So you can think about a very complicated model where your algorithm has a number of dimensions. So the good thing about Bayesian optimization here is that you don't need to restrict yourself to the 2 dimensions that we did. You can have 3-4 dimensions. Mm-hmm. So you could have a task as another dimension. You could model fatigue, for example. The way we're starting it is we are starting with a working memory task that we're very fortunate that Adam collected in about 60 or 80 people. So Adam did this battery of number of working memory tasks. So out of those 12, I could pick the one that showed a nice functional connectivity between front and parietal areas. Mm-hmm. That's reliable across blocks. Mm-hmm. So I have like a good way to start. This could be a meta-analysis. Mm-hmm. This could be your own study, right? Mm-hmm. Which is the case. Mm-hmm. So I start with a particular task that I see that in terms of magnitude of pulse signal, I don't have a lot of variation across 12 minutes of acquisition in a blockwise. Mm-hmm. So the idea is that I start with this and then I impose the brain simulation on top. Mm-hmm. And this is the first step to start with, but you can embed in this other degrees of complexity.
And how do you change the stimulations? Do you then change the stimulation site? Say DLPFC doesn't work for person A. Do you then move it around and try IFG or something else?
Yeah.
Is that how it works? Like
It can. So what we did is to put people with, so they go into the scanner with a particular setting, but you can get people going to the scanner with an array, and what you do digitally in your computer is just to change the combination.
and see which one is
Absolutely.
leading to better performance.
Absolutely.
But then you constantly test the performance.
Yes.
right?
In this case, we constantly test physiologically. The reason that I started with physiological and not behavior is that normally we don't have many tasks that I could think of where you can get a reliable estimate in a short period of time. Whether it's with a physiological signal in 30 seconds, I sort of have some sort of reliable estimate.
But there's also a problem, like if you do it on healthy people, they're already good at the task. I mean, are you going to see an improvement?
So what
You
you
know.
can do is that people are better at tasks, but you can explore, you can make the task always harder and harder, and you can explore that. So in
But then you need a special task which has an adaptive algorithm.
yeah, but that's feasible, right? So what we did
Yeah.
before,
But then
so we have this study in Nature Communications. What we did is we look at large battery of tasks, so 16 different tasks, and we start to see which task or tasks better increases activity in one frontoparietal network versus the other. So we use this as our stage one of the algorithm. We optimize for that. Out of the 16 tasks, we show that actually the convergence doesn't happen in the task that we expected from the meta-analysis, and then we really zoom in into that task and we modulate the difficulty, and we see sort of a quadratic response. So if it gets too hard, your activity doesn't catch up with that, and people kind of they just quit because they can't do it. And this is the same that we did with the phosphine. So we didn't do everything in the same experiment because it becomes intractable in terms of the algorithm figuring this out in a feasible amount of time with the sort of like contrast to noise ratios that we obtain in humans. So what we do is that we first optimize for a particular frequency, and then we optimize for phase. So we know we keep one of them sort of stable, and then you can optimize for another. So you can think about like stacking it or zooming in into different questions.
Thanks for a very nice talk. It seems to me your methods could be adapted for looking at the neurobiology of personality. If you're interested, I can put you in touch with the best personality researcher in Kings. That's me.
Really?
But yeah, it seems like you could even just start off with your percepts thing and just look at individual differences in perception of them, as in terms of like the big five models and things.
Yeah, so I think anything that you have a large parameter space to start with, because not everything is going to be a suitable experiment for Bayesian optimization. Suitable. Everything was suitable, but perhaps not the smartest approach, right? So if you have a clear hypothesis, you can just narrow it down at the hypothesis. So this, what it allows you to do is when you have a lot of possibilities to narrow it down in an efficient way. But yeah, so if in personality there's lots of different possible theories, absolutely.
The room is booked at mine by somebody else, but they're not turned up, so I just have one question. It looks horrendously complicated, because it looks really good. So, I mean, how much of a learning curve are we talking here? I mean,
So, there's lots of people working on it, and we are working on really making accessible. We're making a toolbox.
oh, really? Oh,
Yes.
amazing.
So as part of like this BBSIC project, the thing that we're starting with is really getting all of this separate scripts and not in the best way to give it to someone, and to build a Python toolbox.
Oh, wow.
So that's what we're working right now. So in parallel while we develop, so we're doing this, and yeah, so you do have the expert, and Rob is working on this in different applications here. So I'm working with him in getting this toolbox so we can give it to people to try it out.
Does anybody want to sneak in another question?
Yeah, well, how good is the model at predicting if the function has two peaks? Or predicting like the maximum of the two peaks, or if it just focuses on the first one, then it's fine?
Yes, I think that's really depending on the two things, like your objective function and your acquisition function, right? So whether your acquisition function will be stuck in a local maxima or be able to explore. And this is hard, I would say, to, as you probably know, to predict when you start with, right? So what we tend to do, our approach is actually for a lot of all of these applications that I talked about, what we do is that we do acquire offline data to start with. So we did it for the phosphenes, we did it for the cognitive tasks. So we start by putting just people in a scanner or testing them with these phosphenes in a kind of sort of crazy experiment, if you would think about this, because you sit them in the lab or in the scanner and we test this a bunch of parameters at random, and we try to see if there's an objective function that would allow us to map the space and would allow us to do this in a replicable way. So people do like 3 runs of a particular thing, and then if we have a good idea about the space, we decide on our objective function and our acquisition function. That's always something that you can then check post hoc. It's hard when you're doing it online. You need to make those choices a priori, right? So it might not converge or it might converge in a local maximum.
Sorry, I'm just thinking about the method as a potential interventional method, and how long-lasting learning effects that.
That's a great question.
Yeah.
I wish I had an answer. I don't know. So this mix. So the reason I'm really interested in this is as a way of perturb the brain. So I'm interested in systems. I'm interested in dynamics, and what fascinates me is this just as a way of like sort of pinging the brain or perturbing it, like when you throw a rock to a pond and see what happens. So I haven't played with this because my interest lies much more on the basic side of this, and I don't have a good answer because we don't have good dose response curves or dose response studies. So people have shown that potentially if you stimulate for longer, your effects last for longer. And I've sort of always avoided it. So my first ever study was with brain stimulation was an EEG study to see how short I could stimulate and still see an effect. And that's kind of like we went on the 12 seconds looked kind of okay, and I thought okay maybe 30 for the fMRI. So I don't know.
And just second question. So it's hard to express, but is it the way that you look into it? It's like a physical system where you're tuning into the resonant frequency of the brain, or the natural frequency, and so in the physical system, or
Yeah, so that would be my mathematical framework. So I'm hoping we just put together this project for a PhD student, and I'm hoping that she starts in October looking at exactly building a computational model to have a big computational model with acquiring EEG using graph theory there, so having acquiring DTI to have the weights between different nodes. People have thought about like controllability theory, where particular nodes will influence more your network than other nodes. That's an interesting sort of approach. And the way I want to model is to think, well, we have some sort of like functions. You have neural mass models, and you can think about what would happen if you change the excitatory and inhibitory tone, and this is what tDCS should be doing, where you change the noise, and that's what random noise should be doing, and whether you apply an oscillator, and you can think about like stuff like the Kuromoto model or other oscillators models to model this. So this should give you an idea of then the parameters that you should select and where you should put your simulation. So it's a different approach to this one, but can be combined. But yeah, I don't know. So the idea is that you should perturb in the resonance frequency of the system. So if you think of it as like Arnold tones, that's where you're most likely to cause an effect. If you vary away from that, you won't cause a change because the resonance frequency is very far away. But that on an harmonic, a closed harmonic, makes sense.
Well, I will call it a day. I'm sure people can come up to Ines and ask questions, allow people to slope up. So thank you very much for coming,
Thank you,
and
guys.
see you all next week. Thank you.